Today we discuss about antifungal drugs. The fungus infections are superficial and deep. Both superficial and deep fungal infections are there. Okay. Fungus. Then classification of antifungal drugs. The first one is antifungal antibiotics. Antifungal antibiotics. First one is polyene antifungal antibiotic, polyene antibiotics. Okay, what do you mean by polyene? Polyene means number of double bonds are more. Okay, the example is amphotericin B, nystatin, hemicin, then echinocandins. Okay, then heterocyclic benzofuran. The griseofulvin belongs to benzofuran group. Okay, GPAT question arises like this. Griseofulvin belongs to quinoline ring or benzofuran. You have to write benzofuran. Okay, griseofulvin is an antifungal antibiotic. Okay, then anti-metabolite flucytosine 5-FC, flucytosine, that is anti-metabolite. Azole ring is there. From azoles, first one is imidazole ring is there. Clotrimazole, iconazole, miconazole, oxyconazole, ketoconazole, all these are belongs to imidazole. They have imidazole rings, central ring structure. Then triazoles, the fluconazole, itraconazole, vericonazole. Okay, these are belongs to triazoles. Then allyl amine, terbinafine. Then other topical agents are tornaplate, andesylenic acid, benzoic acid. These are other topical antifungal agents. Okay, we'll discuss one by one. Polyene antibiotics. The name polyene is derived from their highly double bonded structure. Amphotericin B is described as the prototype. Okay, amphotericin B. It is obtained from Streptomyces nodosus. Okay, it is obtained from Streptomyces nodosus. Chemistry and mechanism. The polyene possesses a macrocyclic ring, one side of which has several conjugated double bonds. Okay, and is highly lipophilic, while other side is hydrophilic with many hydroxy groups. A polar amino sugar and a carboxylic acid group are present at one end in some, and they are all insoluble in water and unstable in aqueous medium. Okay, this is the mechanism of action. The polyene have high affinity for ergosterol. Ergosterol is a constituent of bacterial or fungal cell membrane. Ergosterol present in fungal cell membrane. They combine with heat, get inserted into the membrane, and several polyene molecules together orient themselves in such a way to form a micropore. Okay, they will form a micropore. They orient themselves. They are oriented themselves, and they will form the micropore. This is the micropore. The micropore. They will form the micropore. Okay, orient themselves in such a way as to form a micropore. The hydrophilic side forms the interior of the pore. The hydrophilic means water loving. Hydrophobic means water hating. Lipophilic means lipid loving. Yes, philic means loving, phobic means hating. Okay. Then the interior of the pore is hydrophilic in nature. And by which the important ions, amino acids, and other water soluble substances move out from the interior of the fungal cell. Okay, essential amino acids, essential ions, All these are which are present inside the fungal cell, they will removed out by the micropore, because the interior of the micropore is hydrophilic in nature. That's why the water dissolved substances like ions, amino acids, all are coming out from the interior of the cell to the outside. Okay. Then antifungal spectrum. Antifungal spectrum, wide range of yeast and fungi. It will kill wide range of yeast and fungi. For example, Candida albicans, Cryptococcus neoformans. Okay, all type of skin diseases, and nail and skin disorders are due to fungal infections. These are Candida albicans, and Histoplasma capsulatum, Cryptococcus neoformans. These are the different species of candidiasis or fungal infection. Then resistance during the therapy has been noted among Candida. Okay. Then the next antifungal drug, that is nystatin. It is obtained from S. S means, what do you know S? Norsei. S norsei. Streptococcus, not staphylococcus. So that is bacteria. They are bacteria. S norsei, it is similar to AMB in antifungal action. Okay. What is AMB? Antimicrobial agent. IL. CAMB, conventional in formula. Antifungal spectrum, and they have found it. Amphotericin B. AMB is amphotericin B. Okay, amphotericin B. Nystatin obtained from S. norsei. It is similar to amphotericin B in antifungal action and other properties. It is given orally. It is not absorbed. Can be used for monilial diarrhea. Okay. Nystatin is effective but less than, it is very less than azoles. Okay, nystatin is effective, but the effective is less than azole, imidazole and triazoles. Okay, but it is effective against fungal infection in monilial vaginitis. Okay, vaginal antifungal properties. Then candidial resistance to nystatin is not a clinical problem. Okay. Then heterocyclic benzofuran. The griseofulvin belongs to benzofuran group. Okay, benzofuran, heterocyclic benzofuran. Griseofulvin is fungistatic. What is fungistatic? Fungicidal, fungistatic. It will inhibit the growth, not kill the fungus. Okay. It is fungistatic for most dermatophytes, including epidermophyton, trichophyton, microsporum, etc., but not against Candida. The griseofulvin is not effective against candida infection. Okay. Then what is the mechanism of action of griseofulvin? How it will inhibit the growth? How it will inhibit the growth of fungus? Okay, what is the mechanism of action? Griseofulvin interferes with mitosis. Okay, griseofulvin, here the mechanism has been mentioned. Griseofulvin interferes with mitosis. Mitosis means cell division. Somatic cell division, that is known as mitosis. Multinucleated and stunted fungal hypha are produced under its action. It also causes abnormal metaphase configurations. Metaphasic arrest is there. Metaphasic arrest is there. What happens at the metaphase? What happens at the metaphase? From the centrosome, from the centromere, the mitotic spindles, they will attach the center of the chromosome and they contract. By the help of contraction, the chromosomes are separated and the mitotic cell division occurs. It will inhibit the mitotic cell division at the metaphase, or metaphasic arrest will be happen by the help of griseofulvin. Griseofulvin interferes with mitophase. It will arrest the metaphase of mitosis. Okay. Then it does not inhibit the polymerization of tubulin, microtubular protein, which pulls the chromosomes apart, but binds to polymerized microtubules and interfere with their function. Okay. The microtubules are connected with each other and they